Hello everyone, I'm Shanil, an architect and a biodesigner from the Biointegrated Design Lab at UCL. And I'm here to discuss a potential mechanism or a methodology for designing and fabricating large-scale photosynthetic membranes or biologically active membranes for applications within the built environment. Very recently in 2020, a research released by Weizmann Institute highlighted that the anthropomass or the rate and percentage of anthropomass on this planet has increased and is way more than the percentage of biomass. Here, anthropomass is basically everything that is man-made, from cars, roads, buildings, concrete, transportation systems, to plastic and other materials that you find even down to your grocery store. And the biomass is obviously everything that is biologically existing and is grown from the ground up in the planet Earth, including the billions of microorganisms, bacteria, fungi, plants, trees, flora, fauna, animals, everything. And it's actually very, very astonishing to hear or to learn that everything human made is way more now on this planet than what is naturally existing. And if we are to tackle climate change or the problems that we've been facing, that all of us have been maybe experiencing in the different cities that we are dialing in from today, I strongly believe that if we are to sustain and thrive and to sustain life on Earth, we must establish a new kind of relationship with nature, where we do not necessarily demolish and clear out land where natural and biological systems thrive in order to introduce our artificially designed architectural systems, but maybe find a way where we can actually create a dialogue and create a level of coexistence between architecture and biology and natural systems. Now, this is obviously going one step further than the concepts and claims made by the concept of sustainability or green architecture, but we're talking about the concept of bio-integrated architecture, where we bridge the gap between nature and the knowledge we know today available in the scientific field and bridge them with the power of design. Here, we design not just top down in order to, based on the function, the floor area, the FAR ratios, etc., the models that we already know as architects. But the thought process here is to encourage bottom-up thinking, where we're able to bridge the gap between assembly, which is what we do now, as opposed to growth, which is what is actually available or performed by biological systems. Now, this is a very, very important diagram that has evolved over the course of a few years of my research, where that highlights the interdependability of material development or biological material development, fabrication, in this case, the biological fabrication, morphology and form, and it's all integrated at its center with its potential application. Now I'm going to start from the nano and the micro scale, which is going to be a bit different from what we know as nano and micro scale within the field of architecture, but we're going down to the molecular level. We're going to go down to an organismic level wherein humans are not the only clients, but we're also designing to sustain other living organisms. And I'm going to be highlighting or using microalgae for the process and its ability to perform photosynthesis in order to thrive. In 2015, in TU Delft, a very interesting research was published where biomedical experts managed to 3D print using a bioprinter these very, very small centimeter long prototypes, which were printed by a completely biological material, everything that is probably extracted from biological organisms, and created and 3D printed these net-like structure, a crisscross-like pattern on a really tiny nano scale, and they immobilized it with the cells of the microalgae. And over a period of time, what they saw was that the microalgal cells were able to exchange energy and information from its environment, exchange carbon dioxide and oxygen in order to perform photosynthesis and multiply, eventually making the material greener with time. You can see on the right side that they managed to 3D print it, not just one layer, but there are about 25 to 35 and even 50 layers that they experimented. But the scale is very tiny. They were developing to deliver drugs within the human body. So you can obviously understand that this material that they developed, including the microalgae, have the ability to perform as one when embedded within the human body itself. So this material has the ability to adapt to its surrounding environment and actually start exchanging information in real time, as opposed to existing in silo or by itself wherever you embed it. About just 2 years before this, in 2013, Arup in Hamburg created this marvelous structure that has these rectangular glass panels, and these panels are actually photobioreactors, where these are completely liquid, filled with nutrients and microalgal cells that allow the cells to grow. Now the photobioreactors, or these glass chambers, are actually how, in the field of biochemical engineering, they grow microalgae. They either do it in these glass cylinders, or they do it in bags that you sort of see bubbling with nutrient and carbon dioxide and allowing the algae to grow, performing photosynthesis. Now the field of the scientific field of biochemical engineering actually just does it for more biomass, which they then extract and use for cleaning wastewater, producing some parts of energy, or other such applications. And on the left, you're seeing that they've actually embedded this within a material. Now, the constraint with the Arup building on an architectural level is that this is very energy intensive in order to just maintain, let alone extract, the grown microalgal cells. So my hypothesis is that, can we develop materials that can be morphologically designed to optimize the performance of photosynthesis? And can this material have a structural stability, or a structural, have an optimum structural strength, allowing us to at least 3D print or manufacture with this material on an architectural scale? So we started developing multiple experiments with different biomaterials or polymeric materials, such as chitosan, alginates, methyl cellulose, Ghilan gum, xanthan gum, and over quite a few experiments, we managed to actually formulate a series of hydrogels, or water-containing biomaterial or biogels, that when immobilized with microalgal cells, allowed the microalgal cells to grow within the material itself. So here you're actually seeing that this material starts to behave as an environment in itself for the microorganism. Now that is on the micro scale, but if we are, as architects, to begin designing on a larger scale, we need to start formulating geometries and morphologies and a relative fabrication technique that will allow us to create structurally stable membranes or architectural elements for application within our existing environment. So a technique that we started to use, we obviously realized that the water containing gel is actually very viscous. It's gooey, right? And we need this to be gooey if we are to allow the cells to grow. But these microalgal but this material will not have, the viscous material or the gooey material does not have a very, very high structural stability. It only has a limit, right? So we decided to divide our research into 3 different parts. The first was to develop biomembranes, or completely homogeneous membranes from just this material, but varying the polymer concentrations by layer, just like how we see it in nature, on the tree, on the bark of a tree, or the skin, or our skin, or the skin of a plant leaf. And we call this as biomembrane. And then we wanted to develop biohybrids, where we wanted to embed this biomaterial with existing architectural materials. So in order to create biomembranes, taking a lead on from the TU Delft research, we decided to 3D print or additively manufacture with this hydrogel over multiple layers. But before that, we obviously needed to understand its behavior. And along with a lot of physical tests, we conducted rheological properties, rheological characterization in order to understand the flow rate of the material, the curling behavior, its breakage points, its shear stress, and also the impact of extrusion or the pneumatic pressure applied during extrusion on the microalgal cells, in order to see if if the process even complements the biological system we're aiming for. And we did manage to actually 3D print, and what you're seeing here currently on the screen is like a 30 centimeter long acrylic panel, which has been 3D printed down to a resolution of 2 mm with the hydrogel, and there are about 9 to 12 layers that were extruded at this resolution. Our next stage was obviously to embed these layers, but we simultaneously wanted to increase the scale from 30 centimeter to about 1 meter. And we went on to fabricate these panels, these fibrous-like panels, keeping in mind the viscosity of the material, the microalgal demands, the requirements for the microalgae, so you can obviously ask, why did we just not cast it? But we did. But the point is that there needs to be enough surface area to volume ratio in order to optimize the rate of photosynthesis that the microalgae performs. Therefore, we developed and simulated these fibrous patterns depending on the optimum distance between 2 fibers and the structural stability. So the bottommost few layers, which you see are relatively whiter here, were actually printed with a biomaterial or the hydrogel with the least amount of water percentage to increase the amount of structural strength. And no microalgal cells. And as we moved up the layers, we started to extrude with a material with more water percentage and more percentages of microalgal cells immobilized within those fibers. And over a period of time, we did observe these panels to become greener. It was for the first time that such large photosynthetically active 3D printed fibers, fibrous membranes were actually exhibited for over a period of 3 months at the Biotallinn Biennale in Estonia. You can obviously get to read more about this research, which has been published in the Global Challenges. Now, anything living will have a time scale and will have a decay, right? So every material or every particular environment has an optimum limit or a time frame within which the microalgal cells would continue to perform photosynthesis, and after which they will stop because probably they've reached the material's density or they're in lack of nutrients. So we did start to experiment using the same robotic mechanism but with sensors, in order to where it observes where the biomass is less, so that with a syringe it goes and embeds fresh microalgal cells within the material itself. We also wanted to do this very fun experiment in order to understand how the 3D printed panels actually behave once you stop maintaining them. And here you're seeing that the viscous materials are actually beginning to either dissolve and be colonized by bacteria that might be present within its environment. And over another 1 month's period, it really started to decay, dry. So because the material is about 85 to 90% water, the rate of evaporation actually leaves behind a very small amount of the material, which is very easily decayed and dissolved under natural conditions. Then we started to experiment with biohybrids, because the biomembranes had a limit. You can only create those many big panels or big three-dimensional structures with completely homogeneously viscous material or a gooey-like material. So we wanted to create a relationship between the material's viscosity and its photosynthetic properties with existing materials. So Professor Marcus Cruz, my PhD supervisor, was already working on the development of microporous concrete. That is concrete that has the ability to retain a higher amount of moisture. And together we created these cellular automata like cavities within the surface and the three dimension of the cast that we embedded with this material. And the porosity of the MPC concrete actually allowed to extend the lifespan of the algae-laden hydrogel. We continued to explore different geometries, but then also started to explore different applications. And here we are looking at these reaction diffusion algorithms to create multiple pockets for biophotovoltaic, or the ability of microalgal cells to generate energy through movement of electrons. And here we are simply trying to increase the surface areas within a volume so that we can create more cells either in parallel or in series that can then be connected to produce more amounts or optimum amount of energy within a set or within an enclosed space. So what you're seeing here are another very interesting thing with this particular prototype was that we, not just along with the cavities, we also embedded two different types of microalgal cells. So earlier we were just doing Chlorella sorokiniana, and then we also went on to do Chlorella vulgaris in symbiosis. So some of the internal, more dense frames have vulgaris, while the outermost, more open, larger cavities actually have microalgal sorokiniana. And we wanted to actually see if they work in tandem or if there is a difference within the energy. And they actually started to work symbiotically, and the cell, this large 50 centimeter diameter petri dish, which was exhibited at Centre Pompidou, Paris, along with some other prototypes, actually worked as one large energy producing cell, which is very interesting. We then came back to exploring a very, very obvious application of microalgae, which is cleaning polluted water. And taking a lead from the fibrosity, we wanted to create a biohybrid modular component, which here you see in the form of tiles that are then embedded with the microalgae containing hydrogel. And here the movement of flow, the movement of water flow, the movement of photosynthesis, the surface area to volume ratio to keep the hydrogel moist for longer are the parameters that we used in simulating these fibrous patterns. We also scientifically tested the absorption rate or its bioremediation capacity by in the lab, where we observed that cadmium, which is a heavy metal contaminant, when immobilized or when a water containing cadmium was flowing through these tiles containing a specific microalgal species, Phaeodactylome, was able to absorb and reduce the capacity, the percentage of pollutants by 100 times within 30 to 45 minutes. We then moved on to create a scaled up installation for the first time where we brought these tiles and containing the microalgal hydrogel outside into the external environment to observe its behavior under sun and other environmental conditions such as wind and dust, and actually managed to further optimize the movement of water to increase the level of absorption or wastewater remediation with these tiles. So this is version 2.0 of those tiles. Because we observed that the water needs to be better redistributed across the entire panel, we created these central ridge-like places where, when water flows over a larger surface, tends to fall towards the center, and therefore we wanted to hold more amount of material within those central ridges while creating these horizontal niches that would further spread out and allow the water to redistribute as it flows from one layer to the other. With this particular project, we wanted to introduce a relatively novel scientific biological material into the hands of our local craftsmen, to demonstrate that such materials can be incorporated in our today's environment. So what you're seeing here is the tile being fabricated, and this tile was actually exhibited at the Beasley Designs of the Year in Design Museum, and later was obviously explored within our scientific labs, the performance of these materials, of this particular tile. So from starting with a material or a living material homogeneously to starting to embed it with our existing materials, and then a culturally relevant material such as the local clay and a local craftsman and a local manufacturing technique, brings back this sort of loop where we can maybe use our existing tools and techniques, embed them with our old vernacular techniques and our vernacular culturally available systems in order to actually really leapfrog into developing and finding ways of evolving our existing architectural environment into one that performs biological processes such as photosynthesis. So something I want to very, very quickly just talk upon is the multi-layered approach here, right? I went very, very quickly across the slides, but considering the species that goes into the material, the property of the material itself, its life cycle, to the design in terms of its complete larger geometry, its mesoscale geometry, in order to optimize the biological performance. How do we fabricate it across different scales so that it doesn't impact the microorganism on the micro scale? And then its performance and maintenance. Anything that is living needs to have maintenance. This is very, very, very important. And how do we maintain it? Through either embedding a biosensor, which is what we are exploring with the last project I highlighted, and its regeneration or its complete decay, biodegradability under natural. So here you're seeing that my layers actually go from nano, micro, meso, macro, but the development of every individual project has actually gone back and forth across these multiple scales in order to come to the final projects that I'm showing you here today. I very, very strongly believe, I know that this is DCIO and very, very computationally, but I do feel that while a lot of us say that we are in the midst of the fourth digital revolution with AI and new generative techniques, I do very strongly feel that if we really want to make an impact, it has to be a biological revolution that embeds these new digital tools and AI and generative algorithms can actually really help us and benefit how these forms and how these biological processes are designed, fabricated, and then embedded within our existing systems. So I would provoke the audience that this digital revolution has to be a biological one, and that will only happen across multiple scales and geometries. And I think that is why Abel invited me to speak to the brilliant audience here, to sort of give a bit of a spin that if we are to design living mechanisms, and then with new materials that we don't know anything about, we need to incorporate and use the new tools of technology to further empower us in achieving what we are here for. I really thank you very much for inviting me, and I hope you have an incredible rest of the conference. Any questions, please shoot them out to me in my email. Thank you very much.